So, yes, so when I was asked by Axel to give a lecture on something related to astrobiology, it was quite clear that I was going to talk about something within the context of PACE, Protein Artificial Cell Evolution, our project there. But I started to prepare a talk that was sort of the easy talk to give, is to talk about something which is close to what you do research on, because then you actually know what you're talking about. My involvement in this type of project is mostly on the theory side and the simulation side. Well, it's exclusively on the theory and simulation side, and it's not about the actual chemistry, etc., of this protocell. So I started to prepare a talk on these things, and then I sort of realized that that's quite boring, and it's sort of out of context for a person who is sort of not working on simulations of biomolecular systems all the time, and it becomes boring. So I sort of shifted more and more the focus over to the actual project as a whole, and then it became more and more about PACE as a project and about protocells. And in the end, actually, I think I ended up with only talking about this and not mentioning my own research at all. There might be some slide at the end that sort of just briefly mentions what I'm doing, but it also means that I'm not, I mean, it's more of an inspiration talk than actually a talk about something that I'm really competent in. Well, I mean, I do know the story, but I'm not a chemist, so I really do not know much about the chemistry. It's probably the thing that I'm least competent in all of natural science, except geology, perhaps. So that is a problem if there's sort of a discussion, but I would still make an attempt of talking about what we're trying to do here. So the talk will be about protocells and about how we at this point are trying to assemble a protocell in the laboratory. So the idea is to create a chemical system which has lifelike properties, if you wish, in the lab. Well, lifelike properties means that it has the ability to self-replicate and also carry some type of genetic information, which then feeds back into the sort of fitness of the organism, i.e. the replication rate of the organism. You will see that the protocell that we envision is a very, very primitive chemical system compared to contemporary cells. So it's enormously simple, and that's why we are perhaps are able to do this thing. It's not done yet, so there is no protocell at this point, but maybe in a few years there will be. So our work in this project is within the PACE. PACE is an acronym for Programmable Artificial Cell Evolution, as Axel said. And it's one of the integrated projects in the European Union that started in 2004, and it is a 4-year project, so we are sort of heading into the last year of the project. So it's a project on the level of 7.5 million euros approximately, and it's running for 4 years. So there are these integrated projects which involve lots of partners and are considered to be big projects. Big, at least in a basic science sense. It's a small project if you're on the applied side or trying to build some big experimental reactor or something like this. And the goal of the project is actually not so simple. One could perhaps just envision that we say building a protocell, but that's not what the goal of the project is, because it's not realistic to actually have a protocell within the framework, the time frame of the project. So the idea is to explore some possibilities towards how to build a protocell. So the goal is to analyze by modeling and about with experiments the possibilities of implementing a self-reproducing, self-maintaining evolvable chemical system, which is a more technical way of saying that we try to explore various aspects of problems and possibilities that are in the roadmap towards actually building this protocell. And then there are other things which are related, which are more of the sort of application side of what would you do with a protocell if it existed, and that has to do with doing analysis of thinking about what can be done with nano and microscale design, repair, microrobotics, and information processing within this context. The information processing is perhaps a bit surprising, but the base project is actually funded within the Information Technology Society in the 6th Framework Program. So it has a very strong information processing component, and one of the reasons for that is also that it's strongly connected to microfluidic design. So the actual chemical experiments will take place or are taking place on a microfluidic ship. I don't know if you've seen or heard of these. These are small devices like a microchip with small chemical chambers where you're pumping chemicals in one end, and then you can open and close various types of valves, etc. So you can pump chemicals between small chambers on the micro scale, and then you can have small detectors, etc., and feed back information into a computer and open other valves, etc., depending on what you see, and you could put on electric fields on the micro scale and things like that. So you have a micro-controlled chemical environment where these type of protocells are supposed to live. So they're not living in sort of a big test tube where you have shake and mix things. It's sort of a micro-controlled environment on a very, very small scale, which is important to be able to create sort of or artificially design the microenvironment in such a way that you can have this protocell stable, etc. If I want to be sort of popular science about it, you could say that you have protocells on life support in these small microfluidic reactors. And then the idea is to try to make a roadmap for technical realization. These are maybe things that we're most saying just because of political reasons, but and then the last point is about this the interface between the chemical environment and this microfluidic reactor, which is connected to a digital computer. So it has to do with the sort of refining this interface technology where you interface in the digital world with the chemical world on sort of a micro scale. And this is done mostly in Germany at the coordination site in Bochum at the moment. There's something they call the Omega machine, which is this little microchip with a special design. And now, this is just also more administrative political map, but it could be interesting to see the partners of the project. Here's Chalmers, and then we have different types of a number of different partners all over Europe. University of Copenhagen, Economy Institute, for example, and then we have a few other partners in the United States who are not really receiving funding from the European Union, but still participating in the project. And Los Alamos National Lab here actually has a sister project, which is called Protocell Assembly, just like the title of my talk, and it's almost of the same level of funding. So we are trying to do essentially the same thing on both sides of the Atlantics. Collaboration is a bit problematic since Los Alamos is a strange place and a military lab, etc. But large part of the experiments are actually taking place there. Okay, so what is it that we're trying to do? There are various ways one could address the question of creating a minimal cell, if you wish. You can perhaps call the protocell minimal cell, the minimal type of chemical entity that has the possibility of self-replicating and maintaining information. There are bottom-up and top-down approaches, which is sort of the most coarse-grained scale you can look at this. And the top-down approach is rather popular. It's sort of a gene knockout type of idea where you try to reduce an existing cell to a minimal entity. So you try to sort of reduce the chemical material in the cell until it dies. And you try to do that, of course, in a smart pathway so that you get something minimal left that is actually able to survive. That means that it's top down because you start with something that you know is alive. So you sort of start with the success and then you boil yourself down to the minimal thing. And that is currently done in many ways in many places in the world. But that's not what we're doing. We're doing the bottom-up thing, which means that you start with something, you try to build life. So you start with something that is not alive, and then you try to build something that is alive. But of course, here there are many different approaches to that as well. One can start by using autocatalytic RNA, some type of primitive ribosome approach to this problem. That's done in Ricci's lab at Harvard, for example. So they're trying to use existing machinery that is we know is active in the existing cells, like ribosomes, that has sort of key functionalities that are needed to do replication. And ribosomes could, in principle, produce RNA, and they consist of primarily of RNA. So the idea is that they can perhaps be made to produce a material to build themselves, and then have a self-replicating entity. And it, of course, connects very well to this RNA world idea that RNA has this dual functionality, which both can act as both as an enzyme and as an information carrier, and therefore maybe was sort of the first molecule to start acting in a lifelike way on Earth. And then there are other ideas like autocatalytic networks of perhaps proteins or something like that, where you have molecules that can act as enzymes, and then they trigger their enzyme in some other molecule reaction, and then if they sort of start to form a ring, a network with a ring, then there's this ring itself enforcing it can start to self-replicate and be enhanced. And then there are people working on self-replicating lipid aggregates, David Deamer and others, where you have a lipid aggregate that grows and splits and self-divides. And then, of course, you have to connect that to some type of information carrier, so you have to have some type of information molecule that sits in this lipid aggregate and somehow is connected to the chemical metabolism of the lipid aggregate. And then we have the phase project, which is essentially part of this type of effort, where we try to do a lipid aggregate with a connecting information molecule. I will get into the details of that shortly. But I mean, we're not trying to make the argument that our approach is much better than anyone else. I think that these are alternative approaches, and certainly these two, the top-down and bottom-up, they answer completely different questions. So they're not exactly competing in that sense. Down here, you can say that we're competing to create the first protocell from non-living material, but since the approaches are so different, they will probably answer completely different questions. In these approaches, you use molecules that are already evolved. I mean, ribosomes is not something you find out on the moon or anything. I mean, this is something, a highly evolved molecule that is very, very carefully selected for. It's not alive in its own sense, but it's created by something living. Whereas in our case, we want to really start with molecules that are sort of not alive at all. I mean, they're not a product of a living system. This can be argued a little bit about it. But they're not as advanced as a ribosome, at least. But first, I will start briefly by the RNA world. I mean, I guess you have taken that you know about the RNA world, right? Or a little bit. It's not a fair question to say you know about that, right? No, but the idea with the RNA world was that RNA was discovered that RNA in itself could have enzymatic properties, which means they can enhance other reactions. Because many of the functions in our body, or all the functions in our body, but many of the important functions in our body is done by enzymes. Primarily proteins. Proteins are very active. The point of a protein is that it folds itself into a geometric structure, which has the ability to trigger other chemical reactions in various ways. So there are enzymes, and they're very, very important to trigger the different types of metabolic or pathways that are important to the cell. And then it was discovered that RNA actually has this ability as well. It also folds, and it also has an ability to act as an enzyme. But RNA also has the ability of storing information because it has a basis just like DNA. So it can both carry the information in the genome and the actual function of the enzyme. Normally this is done by two completely different molecules in the body. The information is in the DNA, and then that's sort of transcribed over to a protein, and the protein does the actual function that the DNA has encoded for it to do. But RNA can have both of these functions at the same time in one molecule, and there was great excitement about this because that might sort of indicate that, together with the fact that RNA is such an integral part of the whole machinery in contemporary cells, and there are even viruses, of course, that consist only of RNA, was sort of an indication that RNA might be the first molecule around that started the whole origin of life idea. Started in the beginning, or at the origin of life, RNA might be the molecule to bridge the non-living and living matter bridge. But it has turned out to be rather hard to get these RNAs to self-replicate and do much. The naked gene, or I'll use the sort of acronym, the naked gene, which means it's a gene without any context, advanced chemical surrounding, which is doing most of its work, has turned out to be rather hard to realize in a functioning way. There are self-replicating RNAs, molecules. Dieter von Kudowski, for example, have made some. He's involved in the PACE project, one of the experimentalists working on this. But they're very, very specially designed, and they don't seem to be. It's not easy to generalize that idea to say that, oh, this is probably what spontaneously happened. They're not very vulnerable. They can't change very much into something more and more rich or anything. They just do the self-copying in a little spiral, and then they spread out on the surface, for example. And then it has been proven complicated to get this naked gene off the ground. Of course, you can argue that that's just because we don't have enough time and we don't have enough volume to try it. That could be true, we don't know. But it has not been as easy as one hoped in the beginning of the RNA. Just put lots of RNA and then all of a sudden get something that self-replicates out. It wasn't as easy. It doesn't mean that it was a bad idea. It's given huge amounts of information. But then there was this other idea about the lipid world. It's not a different idea, it's just a complementary piece of information in the whole puzzle about origins of life. The idea is that the lipids, like lipids, think about oil or oil lipids, they self-assemble into complex structure completely spontaneously. So, for example, if you have I think one of the first experiments that got people excited here, I think it was Diemer who took meteorites and just essentially they would drive amplifiers on these meteorites, and he poured water on it, and you get self-assembling of complex lipid structures like vesicles and such, just from doing that process. And those look like, they look like something cell-like. Of course, it's just because they have a cell wall. So they have one part of the component which seems to be necessary to make cells, namely the cell wall. They don't contain anything else, but still they do that. They grow and they can divide and they can do all sorts of things. So this is a very complex chemical environment that can be created spontaneously without much detailed control of the whole cell machinery, etc. And the idea is to use this observation as a sort of to understand that this compartmentalization could be used for information-carrying molecules as sort of they can just hitchhike on this spontaneously forming process of lipids that grow and self-divide. And if you get some genetic material that can hitchhike on this, then you have something that starts looking like a cell. So this is an idea that people are following, and we are also following in the footprint of this idea. But to get something lifelike for real, and mind you, I'm not really saying life, because then you get into a discussion of what life is, and I don't want to do that, but saying lifelike, and it means self-replicating and information carrying, and to some extent evolving, even though the evolving part is complicated, because it's not clear if you should ask for open-ended evolution, or if you can sort of say that as soon as something just evolves a little bit, then the pure volume of what could happen in a very long time could lead to open-ended evolution. But in any case, we start to try to do something that can evolve just a little bit and see what goes on. But the parts that needs to go into this story is you need some type of vesicle, protein, some type of container that sort of ensures that the chemicals that are involved in this entity, which is the protocell, stays together and don't just diffuse all over space. It's not absolutely necessary to have this. You might think of a reaction on the surface that sort of stays together without any real compartmentalization, but most of the realization which seems technically feasible has some type of vesicle that carries and keeps the chemicals that are involved in the protocell together. You can certainly not have them spread out all over the place. I mean, you as an object, as your body, needs to stay together, to work together, simply because of how physics works. I mean, it's a local process, right? And then you need some type of metabolism, of course, otherwise thermodynamics will make the whole story very boring. So you have to have some way of using energy sources or entropy sources from the rest of the world. That could be a redox reaction, it could be a photoreaction, it could be various types of things. I was mentioning this because these are the suggestions that we have been using. Of course, photoreactions are quite appealing in many ways. And then you need some type of information-carrying molecule, and then templating molecules are of course perfect for this because they can carry information in the base pairs. So, and you can actually copy and vary the information and do a new copy. So if you do templating, you have this ability of changing something in the chemical, and while it still maintains possible to copy the molecule. Right? And this is the genius of the DNA or the RNA. That it's possible to change the information and still have a molecule that can be copied with the new information. It's not a trivial. If you just ask the question how to do that, it's not easy to find a chemical that will do it. But I mean, Watson and Crick did sort of their genius to realize how you could possibly do such a copying process. How can molecules carry so much information and change that information smoothly between different in a way that life does. So, but you could envision other types of information carrying, like you can carry information in the relative concentration of different chemical species and various types of things, and there are other suggestions here. But we are sticking with the whole templating story. So, are there any questions at that point? Or? So just to clarify one thing also that is very important for the whole story. We're not trying to argue that this is the way that life originated on Earth. Our story is not about recreating life as it was originated on Earth. It's just to create something which is lifelike. It might not at all be the way that it's actually probably not the way that life originated on Earth, but it's an alternative way that it could come about. So it's not the same question as origins of life, even though, of course, it has great...
Oh, you're too careful.
Yeah, but I mean...
You really hope it is, right?
What?
You really hope it is, right?
No. I really do not hope that it is that way. I mean, then you have to argue why we use the PNA molecule. Was that really abundant at that point, etc., etc.? That's not... but to me, it's not the most...
I mean about the specific molecules you use with this.
With the scheme, yes, that's true. The principle, but that's why I think that this constructive approach to origins of life is much more useful than trying to speculate about exactly which molecules fit in where and etc. etc. To me, it sounds more interesting to try to do it, and then as soon as someone does it, you will have 100 different alternatives immediately that can do it in another way, and then after a while you start to build knowledge about this, and then maybe we can answer the question of origins of life, or maybe not. Maybe we'll never answer that question. It was a specific event, after all. We don't know. Maybe it's not the most important question. The important question is that it's possible to bridge this.
Yeah. Will you do a walkthrough for that one?
For this one?
Yeah.
Yes.
Okay.
Yeah. What was there more comments on that discussion? But I agree with Jan. I mean, the idea, the principle is there, but I'm not arguing for the exact molecules. Or we are not. I mean, I am definitely not, because I'm not competent to argue for any molecules.
You wouldn't mind making Frankenstein's monster, right?
You mean which version of the movie do you mean?
A new kind of life has never been seen before. That's what I mean.
Yes, but Frankenstein's monster was pretty close to something that we have seen before. Yes, a little bigger and a little slower. No, but it's true that we sort of more coming from, I mean, Steen, who originated this idea, is coming from the artificial life community, which is definitely working on the question, more on the question life as it could be than life as it is. Yeah. And therefore, this is an alternative life form, if you wish, which is probably not compatible at all. Actually, we wish it not to be compatible with living life as it is now, and that may be for safety reasons, etc. I mean, we don't really want. It's actually a benefit not to use DNA, because it limits the ability for this thing to do nasty things to existing life. So there's definitely a safety issue. Even though, of course, if you get this off the ground, they will not be able to survive very long. But the counter-argument to that is that they're very small, on the other hand, which makes it difficult. I mean, if you have something under 10 to 15 to 20 nanometers, many of the defense systems that we have are not very well adapted to looking for this type of molecules. There are safety issues involved in this. That's definitely true. Not yet, since we don't have anything, but there will be safety issues once this gets off the ground. There are safety issues with the whole nanoscience research in general that I think is not being emphasized enough. When you have things on the nanoscale, they pass through many of the existing biomembranes very easily, and those are nasty things that we don't know how to deal with very well. And I think that should be discussed much more in general by scientists and others who are competent to answer these questions. So we're looking at a new problematic picture of threats from the nanoscience department. Where were we? Yeah, we are trying to create a prototype, right? So I'm stopping by here, but this is the first part. This is actually the easiest part, as I already said, how to make the container. But this is, if you want to be sloppy about it, it's almost like mixing a salad dressing. It's not that hard. You put this lipid in water, as I said, it has to be water background, and then they self-assembly to various types of structures, depending on the length of the tail and the pH and some other well-known chemical properties. You can get micelles, you can get vesicles like this, and you can get other types of phase structures. And these are well known, and you can do some nice theory with differential geometry, or you can simulate them, etc., to see which type of structure you will probably get in this process. So, but the idea is, the important thing is that this is automatic. You don't have to care about this thing. This happens whether you want it or not. The thing that can become a little bit problematic is to get them to split and self-divide in the way that you like. That's not as obvious, but to get them to form is not very hard. And indeed, there are many labs in the world who works with these types of microemulsions, etc. And the cosmetic industry is, of course, very interested in this. There's a commercial going now with micelles that carries this anti-wrinkles cream down in blah blah blah. So if you want to make money, you can always say that you're working for helping them along. But we don't want to do that. So this is the gene, the templating molecule that will carry the information. These are the contemporary information carriers that are used in life as we know it. DNA and RNA, old friends that we know by now. But we are suggesting using another molecule which is very closely related to DNA and RNA. It's called PNA, and it has the same basis. It can carry the same type of basis as RNA and DNA does usually, but it has a different backbone. It has a peptide backbone, so it has a slightly different chemical structure on the backbone of the molecule. And the reason why we want to use this molecule instead of these is that the backbone is hydrophobic, which means that it likes to sit in the lipid aggregate. So when you throw it into water, cartoon chemistry tells you that this thing here will sort of sink into the lipid membrane and sit there. So it will sort of interact naturally in self-assembly together with the lipid aggregate. So it will sort of hitchhike naturally on the lipid aggregate when that as an entity. So it's natural for this to be involved in a very primitive protocell in that sense. I mean, normally the DNA doesn't sit in the membrane; it's sort of contained within the cell. But that's a more complicated thing if you want to copy it, because then you have to coordinate the copying of the and to make sure that the DNA after the splitting actually remains within the cell and not happens to go outside in the water and just go away. But if you have something that actually likes to sit in the membrane, it's easier to ensure that it actually stays within the protocell when it splits, etc. So, and also during the cell cycle. So the connection here, very good resolution. These are pictures that I've stolen from a popular science article that was written a year ago or so in Swedish magazine about our project. So it's not, I haven't done these nice graphics myself. But this is how we envision that the lipid genetic system will connect together. So it's a little bit of what I already said. You have these the lipid aggregate that can grow and divide like this. We'll talk about the growth and why it grows later. Or it can be a micelle, or it could be a vesicle like this. A vesicle is a bimembrane, so it's actually something which it's a bimembrane.
Sorry.
No problem at all.
Swedish Railways are calling you.
But so there's water inside this and outside. So there's an inside and outside. So this looks more like a contemporary cell than this one. This is just a blob of lipid molecules. So they just stick their head groups out into the water and then their tails in a blob. So, and these are very, very small. They are nano 10, 15 nanometers, whereas this can be rather big. It can be on the order of a contemporary cell. So this is something you can see in a microscope. So experimentalists really like these things here because they can see them in the microscope. Whereas theorists like myself really like these things because we can simulate those, but we can't really reach the time scale of these. So there's, but of course the chemists win or the experimentalists win. I mean they set the stage, so we have to adapt and try to simulate these anyway. But these are the two different structures that can be involved. But here, even if we choose even when we work with this vesicle here, the idea is not that the genetic material should be inside; it should still stick in the membrane. So it's the lipid that carries the information. The inside and the outside is not the primary thing. So it's not like a normal cell in that sense. But here is the PNA now. So the PNA sits here, and then the backbone likes the lipid. It's hydrophobic, so it doesn't like the water. So it doesn't like. So it sticks with the backbone into the lipid, but the base pairs they're still hydrophilic, so they like the water, so they stick out. So that's sort of the thermodynamics of that system. At least on a quantum level, of course, you have thermal fluctuations, etc. But on average, this is essentially what happens. But then you have these things floating around, which are building blocks for making new DNA molecules, and so they are floating around in the background where you have to feed them because they will, of course, after a while stick here. So they will come in and sit and sort of base pair here, or they might actually, which is not shown in this picture, stick to the membrane surface and then diffuse around and find something of this order, and then you get a templating process going like this. Here you see it from the side because when you get the base pairing going like this, there's just both of the backbones are hydrophobic, so they want to be in the lipid aggregate, so they fall into the lipid aggregate. You see from the side here. And then this story down here is actually taking place within the lipid membrane. So it sinks, since it has no hydrophilic bases that are free anymore, it sort of sinks into the lipid aggregate, and inside there you try to get it to split. This is actually the hardest part, to get it to split after the templating ligation has happened. And this is a bit problematic. However, the melting temperature of this is actually lower within the lipid environment than it is in water, which helps the cycle a little bit. But you have the temperature cycle to get this to work. So this is sort of one of the critical steps to get it to actually separate. To get it to template is not that hard, but to get it to separate afterwards is harder. Because if it doesn't separate, this is dead here, then nothing else happens, right? So you have to get this step to work. And then after that, you have 2 copies of the genetic material that can do the same thing again. So that is the idea. Now, of course, the thing is that what you have to also produce. Let me see if it's better to go on to the. I can stop it. Talk about this for a while. Yes.
I mean, your containers. I mean, the way you draw it, it doesn't look self-reproducing at all. It just looks as breaking up and reforming no matter how, because it's going to form anyway.
Yes. But I haven't really talked about the growth of the metabolic growth of the container yet. So this is part of this picture. Because the idea is that of course, as you're saying, now as it was there was nothing that grew the container at all. There was no transforming of some chemical into the amplifier that actually makes the container. But here, this is actually the case. We have precursor lipids, which are some type of ester that I don't know much about, but it's a precursor lipid. So this is something which is not yet a building material for the aggregate, but that can be transformed into it by a redox reaction. Here you have precursor PNA. That's essentially what was drawn in the picture before. These are just oligomers, mainly small pieces of the PNA molecule that has to be sort of joined together to become a genetic material, and this is a precursor redox or sensitizer. That's the thing that this is the food essentially. This is the food that can be used in a chemical transformation to actually help the transformation of the precursor lipid and the precursor PNA to become PNA and lipids. So the idea is that this thing goes in to segments. Unfortunately, I don't have the picture exactly what happens there. But here it is. So this is the precursor lipid here, and this is the PNA molecule. So then this thing here can be transformed over to a lipid molecule by bringing it together with by a light sensitizing reaction. But the thing is that this reaction can actually only happen in the presence of a PNA molecule, because it needs the charge transfer property of the PNA molecule to transfer away the charge that happens in the redox reaction. And in that sense, you're sort of coupling the existence of the PNA molecules in the aggregate together with the transformation of precursor lipids into actual lipids that can build that is building material for the aggregate and makes it grow. And then when it grows and grows and grows, it becomes unstable and self-divides. So this is sort of the chemical connection.
But do you really need such a complicated molecule for electron transfer? Like DNA, couldn't you do it with a simpler molecule?
You could definitely do it with a simpler molecule, but then you wouldn't have, because the thing is that the fitness of the entire, and also actually the reaction happens spontaneously without the charge transfer, but it's just that the yield rate is very low, so it's like 6 orders of magnitude higher when you have the PNA molecule. But the idea with the using the PNA molecule as a charge transfer is that that's where fitness enters the picture in one way or another. This is why the PNA molecule is not like a contemporary cell where that's actually used to transcribe some kind of protein that does something in the cell. We can't have such a complicated machinery, so there has to be a function in the PNA molecule that makes this sequence better than this sequence, or some type of a variability. And then the charge transfer property of the PNA molecule depends on the base pairs that are stuck on the PNA molecule, and therefore, since the charge transfer depends on that, the yield rate of the reaction depends on the base pairs, and there is where the fitness enters the picture. So we have a feedback between selecting for the right type of information carrying on the molecule, on the PNA molecule, because it acts actively in the process of creating new lipids, which goes to the protocell.
So is the idea that you, for each lipid that you create, you also polymerize 1 unit of PNA during replication, or?
No.
Because you have a precursor for PNA, and for each lipid you create, you make the precursor for the PNA assembly. Is that it? Or are you making the assembly reaction also?
That is a very good question, because I'm sure. So this is complicated, because I'm sure that that's not the case. However, this picture looks like this is the case. So I don't know how to. Maybe we have changed the reaction somehow, because so
because it would make more sense to have the two freely coupled, the two reactions freely coupled. I think you could make as many lipids as you want to make.
Yes, and that is indeed exactly.
The vesicle, and then your information molecule would not have to have a defined size depending upon the.
No, and that is the current implementation. But I agree that this picture is misleading in the sense that it looks like what you said first is exactly what's happening. But now my limited chemistry knowledge really kicks in, because I don't exactly know how we're doing it. But they are independent at this point. I know that for sure, because we're not really using the chemicals that are on this. I think that the precursor, the lipid precursor, lipid is the same. However, the number of base of cycles here can be varied, but the molecule that is used for the actual in the metabolism has been changed. So there are changes in this chemical scheme. So they are free coupled. So there is no coordination between this in that sense. And that actually created, when that happened, that created a problem. It's a freedom, which is good, but it's also a problem because if you don't have any coordinated replication between the genome and the aggregate, of course it can very easily happen that the aggregate just grows out of grows much quicker than the genome can replicate, or vice versa. And so synchronization is problematic, because in a modern cell, this is very detailed. There's a very detailed control mechanism that ensures that the growing of the cell is coordinated with the actual splitting and copying of the DNA, etc. So there's no such thing as, well, at least not for a healthy cell, there's no such thing as blobbing up without splitting off or whatever, things like that. But in these primitive systems, there is no coordination between the replication of the information carrying molecule and the lipid aggregate. But it turns out that there's a lucky catch here that helps you out, and the reason is that the charge transfer of the PNA molecule is high when they're actually in the ligated state, when they are sticking like this with the group. In that state, they can be used very efficiently as charge transfer. For charge transfers, they can produce new building material. They can produce new lipids in this state. Whereas in this state, of course, they can't really copy themselves because they need to be in this state to be able to catch new PNA molecules to create a state like this again. So there's sort of this going back and forth. But the self-regulating mechanism that happens is that when the PNA molecule is in this state, it creates new lipids, and then the aggregate bloats up, so it becomes larger, but then the effective concentration of PNA molecule within the aggregate gets lower, and then it's actually preferable for this thermodynamically preferable for this molecule to go into this state again. So it self-regulates in that sense, that it's like this, then it grows and the concentration goes down, and then it goes into this state again, and then of course the concentration goes up of PNA molecule because it's templating at that point. So these two things balance each other, and you get spontaneous synchronization simply because of these two different states. There are two different functions of the PNA molecule in these two different states.
What's your solvent?
The background solvent
Yeah.
is water.
Just water.
Yes. Well, I mean, not just water. You have some pH regulating, in some small fraction. Yeah. But it's in principle just water. Yes.
Your precursors are essentially food. That's what you would call food, correct?
Yes. Then together with the redox, with the photosynthesizer, of course.
Yeah, that's
and you can also call
metabolism.
the light food, if you like.
Yes, but that's metabolism.
Yeah. So that's part of the metabolic cycle. As this picture, it's just a picture that tries to demonstrate how the charge transfer works. You have this lipid precursor sitting like this, and then you get UV light in the presence of this light sensitizer here that sits on the PNA molecule. You note that it is in this state when it acts as an enzyme for creating new lipids, and then you get this reaction going, and then you get charge transfer and then at the end you get a new lipid molecule here that can be used. You go like this, and then you get a new lipid molecule out, and then you have grown your aggregate. Of course, this is cartoon chemistry, but there are. I mean, this has actually been demonstrated in the lab. This cycle. So there are results in this.
Could you please pinpoint exactly and explicitly how far you have advanced in reality? Because in the beginning you said the whole scheme doesn't work out yet, but it appears to me that some basic functions have already been making work in the lab. Is that correct? Or is this part of the thing where you can do this in the
Yes,
lab?
this thing here, but it's not using this molecule. But this scheme here, it has been done. Yes. So in principle, I mean, if you want to answer that in detail, I'm not the person to do that, because there would be a chemist who says that this molecule works on this molecule. But in principle, all the different components do work. What is problematic? The main problem at this point is to get the PNA to separate, and you can do that again by increasing the temperature. But when you do that, you also destroy the aggregate. So this is the problem: to make the aggregate stable under temperatures where you can temperature cycle the PNA splitting, and that has not been solved at this point. So therefore, this chemical doesn't work. So that's the simple answer to what the main hurdle is at this point, and that is not a trivial problem. I mean, no one really knows how to solve that. So we don't know how difficult that would be in the end. And of course, there are various types of experiments where you try different amplifiers, etc., different stabilities, and the problem is to have amplifiers that wants to split. I mean, they're not all amplifiers tend to sort of grow and split spontaneously. Some of them just grow and grow and grow and become very large vesicles. And you don't really want that. You want something that's sort of at a moderate size, is sensitive to temperature fluctuation, and actually split off. But then, of course, when they are sensitive to that, then they're also very temperature sensitive and will easily get destroyed with the temperature cycle. So these are two things that work in completely opposite directions. And it's very difficult to make these work at the same time.
Excuse me, can I ask you a question? I mean, this is not at all my field, but there's something in nanosciences, when you talk about charge transfer, you really mean electrons jumping, electrons jumping from one place to the other. After all, the only carriers of charge you have are the electrons and the ions, right, the nuclei. But in this case, in what sense is this a charge transfer? I mean, what is charge in this case?
in the redox, I mean,
It's electrons.
yes.
I mean, there's nothing else but the electron, of course. And the proton, sure. The ions and the electron. But I mean, so in what sense is that you call it a charge transfer? But all the time you're talking about something like something which breaks, see, but so I don't understand what is the electrons jumping from where to where.
So here you have an electron jumping here, right? Essentially, so you create an effective positive charge hole here. And then that hole, if you wish, can be transferred on the backbone back and forth, so that you can redistribute the overall charge of that molecule, which of course is part of the chemical process, over to this end, for example. And then you have effectively, it's not like it's much better to be over here. It's just that you have a larger surface for this to be relaxed into the background water with a relaxation process, where the water then carries off the net charge of the process. If you want a more detailed answer than that, I don't know exactly how these processes work either. But charge transfer essentially means this redistribution of charges over another, or the possibility to to carry either an electron or a hole. I mean, absence of an electron. Okay, so this is just a picture, a cartoon of the entire cycle here. So what sort of suggested to happen. So it's essentially repeating the old story. You start with these molecules here that are not self-assembled. At this point, you put them in water. The idea is that they should self-assemble into some aggregate, and then you have the PNA molecule right there. And this works. Actually, just as a background thing, the PNA molecule was discovered or created by Peter Nilsson at the Polymer Institute in Copenhagen. They're pretty famous for that. And one of the ideas for him was to use this as for various types of treatment, because it has the same basis as DNA. So you can essentially put up a set of bases here, and then think that they can go. This thing can go into a cancer cell, for example, and block the transcript of that cell, whereas it's not something that the cell typically recognizes, so it can sort of go under the radar of the various types of enzymes that would otherwise sort of disassemble this if it was an RNA molecule. So that was one of the original ideas on how to use the PNA. But it turned out that was very, very difficult for the very same reason that we like it, namely that this PNA molecule likes to sit in the membrane. So it never really enters into the cell core, and doesn't therefore not interact very well with the DNA molecule. But this is also the reason why we like it so much, because we want it to sit in the membrane. So this is sort of the story when Peter Nilsson gave this talk, and Steve happened to be in the audience, and he talked about this problem, and he said, well, wait a minute, that's exactly what I want. So that's why we started to use this molecule. And then the idea is that then you feed this with the resources that we were talking about before, and then it's supposed to grow, and then at some point it's supposed to split like this, just because of the instability that's created when this grows. If you have the right amplifiers, we do this continuously, and then we have two of these. And of course there will be some statistical fluxes here, which means that sometimes one of these will end up without any PNA material in it, and sometimes it will end up with too much. But statistically you have the possibility of creating new protocells with the same P material again. Yes.
Is it clear that I understand what you're talking about? If you just take oil and water and you shake it, you get as far as 2.
Yes, well, not really oil. It's well, the amplifier. Then you get.
That's very.
Yes.
Okay.
Yes, and then you need to have these specially designed molecules to be able to actually micro grow this structure. Yes.
So for the step 3, no light is required. You can already grow the cell. I mean, grow the volume without light energy.
Yeah, I think that no. But I mean, the growing here is, I think, sort of artificial in the sense that this is growing just by loading in this.
Yes.
So this is in the picture. You're loading in the building material. So this is like eating. but you haven't transformed any of these things into real building material. And then you shine light on it, and then you transform into real lipids and new PNA molecules, and therefore, and then you can split. So this is just the loading of the molecule, if you wish.
I think you said that you use ultraviolet light. Is that correct?
I didn't say that, but I think we do, yes.
I think you said earlier, not now.
Did I?
I think you said it earlier.
Oh.
Which surprised me a little bit because ultraviolet would typically totally decompose molecules. And, how does that work? Why do you have to go to such short packages?
Oh, well, that's a good question.
That's one.
Yeah. I mean, of course, the trivial answer is that it depends on the light synthesizer you use, and then you ask, why don't you use a light synthesizer that can absorb infrared?
I was just wondering if it wouldn't tend to destroy your...
No, yes, you're absolutely right. This is correct. So the reason why I said ultraviolet is that I tend to remember that this is again something, I haven't seen this myself, so I don't know. But I think they said they used ultraviolet light last time, and the reason for that is unknown to me. But surely, it's probably, so it has to do with which chemicals you would like to use because of other reasons. I mean, you want it to sit on the DNA molecule, you want the charge transport to work, etc. So, but they do have it stable, do have this aggregate stable, and I don't quite know how I agree with you that the ultraviolet light would destroy the aggregate. Probably, if they're not too quick.
Maybe you want to have some free charger if you want to ionize this stuff, so at least partially for the chemistry.
Like I don't ionize with UV.
Will the UV light?
You will certainly destroy the aggregate before you ionize some sort of spontaneously ionized thing, so that would be a very strong light.
It will be lightly ionized.
But we're not talking about vacuum ultraviolet, but it's not like it's maybe down to 2000 angstrom. That will ionize.
But okay, so this is a speculation on the trajectory, which is something I just heard last summer. They said ultraviolet, and then I probably repeated that. That was unfortunate. Maybe we don't use ultraviolet, so maybe it's not even a discussion. These light synthesizer experiments are done at the Samos, and I haven't been there in a while, so I don't know what they're using. Because you can also imagine not using light at all, but actually using some redox reaction. Essentially. But I think that in the end we decided that light was easier because I think that the technology is just simply more developed in that area. It's easier to ensure that you don't get side reactions that you don't want to have with the light synthesizer essentially.
But normal photosynthesis doesn't work with infrared. It only works with up a certain critical wavelengths, right? So obviously you have limitations there. Yeah.
But that's still visible light. There are.
Yeah, it's still visible, but it's not in the red anywhere. It's near the.
But there are light synthesizers that do work in infrared.
No, I don't know if any.
Oh, aren't they? Okay, so this shows my ignorance again. I'm not sure about this.
So maybe there are different photosynthesizers simply available, and the more interesting ones are for the shorter wavelengths. So what is a sensitizer actually?
Synthesizer?
Sensitizer.
Sensitizer. Sensitizer. Yeah, this is the thing that can absorb the light and transform on the light.
So it's actually the photosynthesizer.
Yes.
Synthesizer then, of course.
So you don't use redox at all in this scheme. You just generate the charge transfers through the UV or visible order.
Yes, but the reaction is a redox reaction still, right? That happens when you light.
It's not chemically.
No, it's not chemically induced in this picture. No. But you could imagine that you use some chemically induced redox reaction instead. That was sort of one of the paths that were investigated in the beginning. Well, I should definitely wrap up here. So That's it. So I should stop now.
Maybe another 5 to 10 minutes would be okay.
5 minutes.
Unless people have to go. We'll go briefly through
I
that.
won't really say much. But I sort of need to say something about what I do know, because it's embarrassing to get the rest of the talk about things that I don't know very much about. But what I do, and our group does in this project, is to do simulations of biomolecular systems. So we try to answer questions like stability questions about of the lipid aggregates. How do the and how do they actually, the PNA molecule actually interact with the lipid, with the amplifiers in the lipid, and try to do simulations of that. And the problem is that, I mean, the easy way, or the traditional way of trying to address that question is by directly doing molecular dynamics. But and even that is actually problematic, because it's not really the effective potentials are not. They don't always do what the experimentalist tells you that is actually happening in the experiment. But even so, the size of these systems are such, and the timescales are such, that molecular dynamics cannot really cut it. I mean, you can't really reach self-assembly timescales and to study the dynamics of the vesicle, definitely not the vesicle, but not even my cell very well if you use molecular dynamics. It's simply
what is the timescale?
The timescale is milliseconds essentially that you would like to get to, and you are orders of magnitude of them. Maybe 6 or so orders of magnitude at least of that. So you need to do some type of coarse graining. So and the question is, what type of coarse graining do you want to use? And then that's where we do our work. And there, so to develop these types of schemes, I won't go. I mean, this is just a sketch of the different types of timescales you would like to address things on. It goes from actual quantum mechanical simulations all the way up to evolution of functionality here. And we are typically working on this here, and on this here, and this here. These are the different things that we are working on. And we have been working with a technique called dissipative particle dynamics, which novel is maybe, you should take that away. It's becoming rather standard now, so it's not very novel anymore. And we did not invent it, by the way. So it's just a way of thinking about mesoscopic simulation, where you try to think of, where you try to simulate not particles, but actually clusters of particles, and then you immediately end up with something, some dynamics of a denser band type. This is, of course, standard knowledge that when you try to project, in principle, a deterministic dynamical system, molecular dynamics code onto some aggregate level, you typically get part of the dynamics become deterministic, but also you get a stochastic and a dissipative part. And this is a way of dissipative particle dynamics. It's a way of capturing the dissipative and the stochastic driving in a way that you still conserve momentum and angular momentum and mass in the system. And then you get something which is consistent with Navier-Stokes, of course, just because of these conservation laws. But it can also capture, for example, these molecular self-assembly processes rather well, depending on how you see it. Because there are various types of problems with this, and that's really where we do come in. We try to improve this scheme, because this is rather good at capturing the equilibrium of the system, but the dynamics is not very well captured in these schemes. You can actually show that you have all sorts of problems with different timescales and Smith numbers and other things that just don't fit with the experiments. And it seems to be very hard to actually do mesoscopic simulation in a way that is consistent in all aspects with the hydrodynamics in that sense. But what we're trying to do, just to finalize, is to use inverse methods, so that we run the simulation in molecular dynamics in full scale, but very, very small and for a rather short period of time. And then we try to look at this data, and through an inverse problem, we actually try to determine exactly how the functional form of the types of interactions that needs to go into the Lange-Band type dynamics should look like. And that's it's a bit technical to actually separate the deterministic and the stochastic part, and etc., etc. But there's a technique to do that. So that's what we're trying to do. And then when you get these parameters, you can plug them into a large scale sort of coarse grain dynamics that you can run. And then at some point you need to recalibrate, because the calibrations are done at equilibrium, and you maybe have to recalibrate the system. But in that way you can actually reach timescales which are much longer than you can do with molecular dynamics. So that's what we're trying to do.
This large event, I mean, I know this expression large event rates, for example, in ion molecule reactions. Is there any resemblance?
Well, that's because, I think that's a way from a stochastically driven system to jump over potential barriers, right? So the stochastically driven...
calculating the cross-section.
Is the probability the same?
Yes, but so the stochastic dissipation is a way to capture the temperature dependence on pumping the system, right? You need that, otherwise you won't tunnel through anything, classically. So it's that's. But the Langevin equation basically is just a way to say that you have pumping and dissipation, and those are of course related through the dissipation fluctuation theorem, and it's a way of capturing that type of dynamics where it's an effect of hiding lots of fast degrees of freedom into sort of black box, and it looks like noise and dissipation. It's done by the sort of Schwarzschild Mori theory or projection operators, etc. But this can be done only in theory, and it's really hard to actually do any of these calculations on a large scale. Therefore, you have to find other tricks on how to actually estimate how these rates and the functional form of this dissipation, etc., should look like. But you can do that by solving this inverse problem. And the reason why you can do it is that the problem is separated. The deterministic part is directly given by the pair correlation function, and it's not affected at all by the pumping and the dissipation. This is an old theorem by Henderson that there's a one-to-one corresponding between the pair correlation in molecular in a multi-body system and the actual pair potential, conservative pair potential at equilibrium. So that can take out that part, and then you have to measure the stochastic and driving, and that you can do in another way. So, but if you're interested in those details, we can have a discussion later on. I don't want to keep everybody online for that.
You have your conclusion, or is that here?
Well, I have, but I don't want to go into more slides, so let's just leave it there. And so
okay.
This was the aim of this was to give some hint of or inspiration talk or whatever on a project which is going on to create protocells, and we will probably have protocells. I don't know if we will do them, but someone will do protocells or something protocell-like within the next 5 or 10 years. I'm pretty sure. If I'm not going to be conservative, I'll actually stick my neck out a little bit. And that will open up a new avenue of both technology and knowledge, which will be very interesting to follow, I think. So you've seen a glimpse of that.
Are there any other groups in Sweden working on similar projects?
Not that I'm aware of, no. Not in Sweden. No.
And abroad, you showed on this collection of universities and networks.
Yes, but that was only within the PACE project. And we have the primary players here. I mean, there are many groups that are doing similar things, especially doing lipid chemistry. But the main players are Ricci's lab at Harvard, I think. They are very advanced. I mean, they will, and will probably, yeah. I will put my money on them in one way or another. And then there is David Deemer, who is currently, is he in Italy? I don't. He's moved around, but he worked on the lipid world for a long time. So he's also trying to do protocells now. But I mean, there are an increasing number of groups that wants to do this, and of course it's in the air that this will be done. So an increasing number of people saying that they're doing this type of science. But I think that the primary players are Los Amos and the PACE project and these two groups that are actually really doing protocells. Otherwise, other people are doing aspects of it or trying to say that they're doing it because it sounds more sexy to than the actual research. But so that's at least that I'm aware. But I don't know. I think that there is a group in Japan also. That I don't know exactly which one, but that are doing similar things. Maybe it's Takashi's group.
So I will now ask for some questions, but let's first of all thank Martin for his very interesting talk. So, questions please. Pille.
Yeah, I wanted to ask you a little bit about